I will explain the steps now one by one. Okay, here this is the overall summary of the homology modeling technique. First one we see here, this is the structure we need. So, we have the two sequences. So, identify the template, right, we check the alignment and get the template. Once we get the template, then go with the alignment corrections, right, here with the this is the alignment, right, and we check the alignment and see so that there will be less gaps in the alignment. Once we have the alignment correction, then we generate the backbones, right, because we take the same backbone and get the backbone, and then when the backbone is set, then look for the loops. Okay, here so you can see several loops here and model these loops, right. When the loops are done, then go with the side chains. There are several libraries, you can check the side chains, and when we model the side chain, then finally optimize the model, right. You can also try to use molecular dynamic simulations to see whether there is any fluctuations in these amino acid residues, and finally validate your model. If you are happy with the model and it satisfies the requirements, then it is fine. If not, you have to iterate again and again with the different steps. We change the loops and you change the template or change the side chain, right, and finally see whether the model is valid or not. First we need to take the template. How to get the templates? You check with the blast, your sequence you give and run blast, and finally you will get lot of hits with the different identities. So, your accuracy of the model depends on the template, and again it is very important to identify the best template. If you get several hits with high sequence identity, what other criteria you use? You can see the sequence coverage and you can see the less number of gaps and see whether the proteins belong to similar families, right, and all the aspects you need to consider, right, and second is see if these proteins have some similar folds and the regions are properly aligned. So, all these things you need to consider before you select the template. Then the second one, you look at these different proteins and see the important residues, whether the residues are the same in these two different proteins. Then based on all these aspects, then you can choose a template, because it is very important to choose a template, otherwise you end up with the wrong structure. So, now if you see the structures are known, because you search your query with the known structures. So, then we get some structural guidelines with the templates, you will get different structural information, because the PDB is known, and see whether you can see the family of the proteins, how about the structurally conserved regions, and then if you see all these informations, then you can choose the template. How do you check the conserved regions? Because these regions, what are conserved regions? On the protein they have the nearly identical structures, right, because the conserved regions they have the identical structures, and there might be the interior core and mostly they contains alpha helix and beta strands. So, look into these details, right, and also location of these residues, right, in the conserved regions, because we know the structures, right, we need to take into consideration all these aspects. Then we use the biological information and the known functional annotation, because in several databases you can see the residues different functions, right, other active sites or motifs and so on. But using all these information, finally you can choose the template. Usually what we do, you take a sequence and just paste the sequence in Swiss model or any software, you will get the structure. So, the question is whether you get the reliable structure or not. So, if you spend some time, right, in each steps, then you can confident that you get the good structures. The most important one is a template, you need to give proper template for the modeling. In that case, you can get at least more than 60% confident that you can get the reliable structure. So, this is the method we use for the taking the template. You use the BLAST and then you use the known structure database, right, and finally you get the structures from the PDB, right, this is fine. Okay, now what to do? You get the template, right, you have your query sequence and you get the template, you check the literature and other information, finally you decided that okay, this is your template. Okay, then if you see the alignment is correct or not. For example, if you see an alignment, right, alanine is mutated to glutamic acid, So, you can align any residue, you can mutate any residues, that is possible, but this is reliable or not, right. If it is alanine is the buried region and we convert the mutated to glutamic acid, this is what unlikely happen because it will destabilize the protein, right, because alanine is a hydrophobic residue, this is hydrophobic core, and if you convert mutate alanine to glutamic acid, what will happen? Destabilize, right, because it is a charged residue, right, it will destabilize the protein. So, this is less possible to have this type of alignment. In this case the alignment you need to change, so introduce a gap or right, and you can make these residues not aligned, you have to align properly. Then you can use a multiple sequence alignment and see that conserved residues and your template whether the conserved residues are properly aligned or not. If it is not properly aligned, you have to make the correction so that it is properly aligned. Then you can see the template and see whether the core residues which are less likely to be changed, because these are the very important residues, right, then the residues are the outside of the protein. Then also you can see the insertion deletions and the gaps, right, you have to make the gaps as less as possible, because if you introduce more number of gaps it is less possible, right. In this case you check that the gap should be as small as possible. Okay, I will show an example, right. Here you can see a template structure, this in green, okay, this is the green one, okay, this is the template structure. So, we aligned with two types of structures, right, one is you can see in red and one is in blue. If you see the red one, okay, this is aligned here, this is the red, and here this is up to here, and this is the gap, okay, here this is the large gap if you use the red one, and there is another one you can make the corrections using the blue line. You can see the blue is up to here and again it goes from here, and this is the gap, this is the gap for the one alignment, this is the gap for the second alignment. In this case which one you have to choose? The second one, right. In this case you can align with less gaps. Then if you now you have the template ready and you change the corrections, alignment corrections, and the alignment is also fine, right. Now what is next step?
Build the
Backbone generation, right, because we have the sequence, like for example you have the sequence, right, and this is your query sequence, and you have the template, you get the template, right, for example you have the template. Then if you see this one, so backbone is the same, because if you draw this, right, this sequence you can get this one, right, n C alpha C n C alpha C n. So, here you have the hydrogen and this R group. If you take the query sequence or the template, the backbone is the same. What is the difference? Different side chain. For the take the first one, you can see A I and it goes on, and second one A I R. So, backbone is the same. In this case you can just copy the backbone, and also if you see some cases the side chains are the same. In this case you can also copy the side chains, right, this is how you can generate the backbones. Okay, see this is the backbone, right, of your template, right, the backbone of the template. For your target you copy the same, right. Now for your target, right, you just copy the backbone, that is fine, and here what do you do? So, use the same backbone, and here you can see some side chains. What resembles this side chain?
Similar.
These side chains are the same between the template and the target. So, same amino acid residue, for example as I showed here, this A and I, they are same. So, this case you can just copy the side chains, and then next is go with the loop modeling, because this is the important step, because the loops are the basis of the steric overlaps. Sometimes we have the small loops, some we have the longer loops, right. If you take the helices strands and the conformation, they are restricted, right. Helices take some specific conformation, right. What is the range in the Ramachandran plot?
Something minus.
That's minus 50, minus 6, that range, right. You can see the conformation restricted, even the strands are restricted, but the loop they can take any conformation. So, in this case they are highly flexible, it takes different conformation. So, in this case you can see a specific degree of overlap, right, between these different loop regions. Then you have to check the atoms, right, within the loops, and see how these atoms fit in the protein. Okay, I will show an example. So, here this is the actual loop, right. If you take the template and your target, you may see the longer ones. So, you can see this region here, the target you can see a longer loop than the one which are in the your template. So, in this case you need to model these loops, replace these residues. There are various options to loop the modeling. Either you can check the same residues in different protein structures which are all set up in the loop regions, and then see you can try to connect with that residues, and finally you can make the corrections and energy minimizations and to make the conformational changes. So, likewise this loop modeling is an important one because it is highly flexible, right. In this case you can get the data from these known structures in loop regions, and you can make these loops, right, the conformation of this particular loops. So, now we have done the template, we made the alignment correction and we generated the backbone and we fit the loop regions, but loop regions are not highly stable, right, but we made these loops, right, within these regular secondary structures. Now, we have to change the side chains, right. If the sequence identity is very high, then it is mostly done. For example, if it is 90%, right, the 90% of this chain side chain we can already generated, and only 10% of this protein we need to generate side chain. This is the reason why we require the high sequence identity or similarity. In this case, almost the conformation is almost fixed. So, now for the rest of the protein, where the amino acids are different, right, then we need to replace the side chains. We do the mutations and change the side chains. In this case, we need to check the conformation of the side chain. For example, what makes the conformation of the different chains, right? So, you the bond length, you have the bond angle and the torsion angles phi and psi, right, because that is merely from the main chain, and the chi angles. So, because if you go with the side chains, right, it is important to say the side chain conformation. So, we look into the side chains, many residues have more degrees of freedom, right. For example, if you have this alanine, how many rotations are possible? In the main chain you can see one CH2 group, so you can have the rotations. If you go for the serine or if you go the threonine, you can see depending upon this atom the type, right, you can see different rotatable bonds. So, it can take different types of takes different conformation, right. So, in this case, if you take this is a protein, right, here I show the tyrosine 22, it has various conformations. You check the all possible conformations, and then if you see the libraries and check the one which one is the most probable one based on the type of the specific residue and where it is located. So, whether it is in helix or whether it is strand or whether it is buried or exposed, right, you can see different conformations and you can fix these conformations for each side chains. Now, we have the backbone and the side chains are the fixed if they are properly aligned with the same residues, and if the residues are different, then we mutate the residues, right, and you can build this model based on these available libraries. So, in this case it is very important because we need the libraries, right, because if you have the rotations, how much degrees one can rotate? 360 degrees you can rotate, but if you do it different angles and different rotations, it is comparatively very expensive because it searches for different conformations. So, for this case you check for the available libraries, right, there are several rotamer libraries available. How they develop the libraries?
From the existing,
right, from this distance and also from the known structures, right. If there is alanine or there is a valine, right, you can see what is the probable conformation each amino acid can adopt, and also depending upon these neighboring residues or the 3 different residues. So, all these are probable libraries. In this case, if you find your residues, right, in different secondary structures and the different locations, you can get the proper libraries from the rotamers and you can use that conformation to build your model. Otherwise, if you do systematic sampling, it takes long time and it is very expensive based on the computation time. So, we use the libraries and then see which conformation each side chain can adopt, because each side chain can adopt only a small conformation, right, then all possible conformations. You can check with the environment and you can use the particular conformation for any side chain. So, now if you look into this data available in the literature, In homologous proteins mainly they retain the same rotameric state, and if you take the chi values for 80% of the identical and the 75% of the mutated residues, they have same conformation, right, they do not change much. So, if we look into this original structure, template structure and the target structure, the difference of the conformation is less, right, more than 70% of the residues they have similar conformations. So, now you have the almost similar structures. For example, if you have the template and you have the backbone and you have the side chain, you have the loops. So, you have the crude model, right. Now, what we need to see during these steps, we introduced several artifacts. What are the various artifacts we used? We just replaced several amino acid residues, for example, large side chains for small ones, alanine to valine, right, just we put it, right, whether they are sterically allowed or not, that we do not know, we have not checked. And the second one, we take some peptides from different reference proteins like loop modeling, right. If you do not get the proper data, we take from different structures, we do not check whether this is the similar proteins or different and the similar folds or not, we take from different proteins. Then also we do not know the conformation loops, loops can take any conformation, right. In this case we use whatever available in known structures. So, how to rectify this problem, right, we need to optimize it. So, in this case you can do the energy minimization, right, and see whether this protein or this model is chemically and conformationally reasonable or not. So, there are different methods available to minimize energies, right, and then use the energy minimization technique to see whether your protein is reasonably good, energetically favorable or not. Then also we can see the MD dynamic simulations, you see whether what are the possible conformations your protein can visit during different period of time, right. This can also help to remove the large errors, also it can have the low energy conformation. So, you can see this is the backbone, and if you do the dynamics, finally you will get the stable 3 dimensional structure. So, once we develop a model, it is a completely crude model, you need to do energy minimization to see and get the energetically favorable structure, and for more aspect, if you do the MD simulations, you can see what are the probable conformations each residue can make and whether it is possible to better conformation with lowest energy state. So, now if you model the structure and you optimized it and you got the structure, now your structure is done, right. Then the next question is whether this model is reliable or not. We do all the experiments, we check the proper template and minimize the energy and you have done everything, and whether finally they are going to validate. How to validate this? First we see whether the bond length, bond angles and torsion angles, right, all the stereochemical properties they are properly set or not. And the second one, whether the specific important residues, for example active site residues or any antigenic site residues. So, these residues have the proper conformation, right. If for example if you have the active site residues, right, they tend to interact with the other molecules, you should have the some type of pockets and so on, right. You need to see whether all these things are correct or not, and check the correct fold, whether it is properly folded, because if you see the sequences you can assign the fold and final structure is in proper fold or not. There are various methods available in the literature to validate the structure based on the stereochemical properties or based on the energy, right, and based on the experimental information, right. There are various methods available in the literature to validate whether your model is correct or not. So, here for example I give an example, for example ProCheck or this what if, this will check the stereochemical conformations, for example bond length, bond angle, torsion angles and the atomic contacts, whether the proper contacts between different amino acids or not. Then second one is the Ramanson plot, this is also one of the major components for evaluating your structure, whether the structure is mainly in the allowed region or not, right. Once you do this, it ensures that the backbone conformation of the model is normal, because if the most of the residues are in within the allowed region of the Ramanson plot, then you can see that the backbone conformation is almost correct, right. You can use several servers to get the model, but even then you have to check and validate the specific models. So, we see the Ramanson plot, then if you have the template and the query, you will get almost similar. Why it is similar? Because backbone we are just copying from this template. So, in this case almost you can see the similar structures, but during the minimization we change the conformation and finally you need to check whether they are properly aligned. So, you can see this is a template gives a good structure, and you can see most of this the residues which are left side of the Ramachandran plot for the alpha helices as well as for the beta strands. Then you check the specific residues, for example glycine, because glycine has different conformation in the Ramachandran plot that I will show now, and the proline, where the proline are located in the Ramachandran plot. So, here is the plot for the general case, right, where is this your alpha helix, and you can see the beta strand here, and some cases you can see the glycine, this is for the glycine, right, and here this is for the proline. So, check whether the glycines or proline they are also right placed properly, and they have the conformational angles which can be seen from this Ramachandran plot. So, this is one example you get from the ProCheck, right, you get a structure and you submit your structure in the ProCheck server, it will identify the bond lengths, bond angle and torsion angle, and how many of them are within the limit. For example, we see the bond length 99.1% are within the limit. So, these are bond angles, you can see the 98.1%, and the planar groups the 100% are within the limit. So, in this case the model is reasonably good, right, fine. Then see the Ramachandran plot, you can see there use the allowed region, right, these are the allowed region, and you can see the partially allowed region. So, these are the fine around 98% which are in the allowed regions. In this case the model works fine. Now, I give you one example, right, as a case study. So, I have one sequence, okay, this is the tyrosine kinase protein CS kinase, this is important for the target as a colorectal cancer, right. So, to develop a model, right, if first what you have to do, to find the template. So, what we have to do blast, right, first we blast. So, we will get different sequences of different identities, say 90%, 98%, 72% of different identities. Then how to choose your template? First you see the same family and you see the complete structure is known. Sometimes you get the 98% sequence identity, but the structure is not complete, you have only the partial structure. So, you should have the complete structure for this the PDB. Sequence wise align until it is 98%, right, but if you see the structure you have only partial structure. So, you need to make sure that the complete structure is available. So, in this case if we have the human kinase, we choose the 2 SRC, they belong the same kinase family, and this is CS kinase, and here you can see this SRC kinase. And then look at the resolution, what is the resolution of the template you choose, right, there should be a high resolution, right, not the low resolution structures. So, it is 1.5 angstrom, so it is fine. And another aspect is, even if you get less identical templates, you see the structurally similar or not, right, that is also possible. For example, if you have these binding sites, so sites are properly aligned, that regions are properly aligned, are there any specific motifs? So, see that specific motifs that they are properly aligned, that way also even the sequence identity is less, you can choose the structures. In this case your structure will be similar, resemble the functionally important regions, that is also possible, fine. So, if you take this structure, right, here this has the identity of 84%, right, of the positive is 92%, right, here yeah, this is similarity, and there is no gap because all residues are properly aligned, and we have the complete structure. So, with 84% sequence identity, and you can treat this as your template. When you make a template, then finally model the structure, right, like the loop modeling, and you can use the backbone generation and the side chain modeling, right. Finally, you get the energy minimized structure, this is a structure. You can see the blue one, okay, this is for the CS kinase, and the grey one, this is called SRC kinase, and the RMS is around 1 angstrom. So, it is a reasonably good structure, we can get this one. Then we validate our model, this is one of the examples you can do with the Ramachandran plot. You can see the dots, right, which are mainly in the alpha helices and in the beta strands. And we see about 90% of these residues are in the allowed region, and about 9% are in the partially allowed region, right. In this case you can see the model is reasonably good, and you can trust this model for the other applications. So, here the sequence identity is very high. So, you can see the RMS is 1 angstrom. In this case we can also use this for understanding the different sites as well as use this model for identifying the lead compounds in structure based drug design. It is possible because it gives similarly high resolution structure similar to the structures which you can obtain from experiments. So, these are the different servers for homology modeling that you can use Swiss model or the what if different servers you can do it, and here this is the one of the widely used tools like modeler. Is a various options to do that. So, you can download this software, you can install this software and you can get the structures using homology modeling. So, now we did this homology modeling. If you do not have any sequence identity, then what we have to do? We have to do from the scratch, right. In this case we have any sequence, you can get the structure, but we need to do from the sequence based on physical principles. What are the bond length, bond angle, torsion angle, and what types of interactions it can make, 1,4 interactions, 1,5 interactions, right, different types of van der Waals interactions, right, do all these things. It is time demanding because computationally expensive. Why is computationally expensive? Because we need to do from the scratch and it is mainly based on the physical models, right. We need to calculate the energy, we need to fix the different positions for each atom, right. We have to do a lot of confirmation sampling. So, it takes lot of time to get a structure, and this is the reason the ab initio prediction we can use only for small proteins, for example less than 100 residues. If it is very high proteins, because it is very difficult to do all the minimization, in this case what they do, so they cut into pieces based on the domain information and model the proteins for each domains, and then they combine everything together and do the minimization to get the final structure. So, because in the knowledge based approach it may fail because the homologous structures are not available, and if there is new folds because they look for these similar folds, right, to do this knowledge based approach. So, Rosetta is one of the widely used methods for the ab initio modeling, right. So, what they do, they start from the beginning and currently they implemented the homology modeling along with ab initio, right. They take some fragments, right, based on the local sequence preference, and you can see the library of the fragments for the different cases, and they think that these have the native like globular properties. Then they combine all these things together from the populations, and for the small, for the unknown cases they do the modeling, and then finally they generate the model and finally go with the different the possible structures and select the best one. This is how they use. Even IIT Madras, IIT Delhi, Professor Jairam developed a server called Bhagirat, that is also based on the ab initio modeling technique. So, it is also have reasonably good accuracy if the residues are less than 100 residues, if the protein is small in size. So, there is another method that is called the threading or the fold recognition. In this case you can see the best fit between the sequence and template. For example, if you have a sequence, from the sequence we can derive some information, we have a energy function and see how this can fit with the structures. This is a kind of 1D to 3D alignment. So, what to do? We first get the energy function, that is very important, that is a difficult step, and template libraries. You can see the different structures available in protein data bank. You make a libraries and see which energy function fits with which type of proteins, right, and then align the sequence with the template using score. The score they depend on the statistical potentials, how far the residues are close to each other, or what is the environment based on physical chemical properties, and how about the gap. Based on that they develop the energy functions and they compare, right. This is the kind of 1D to 3D alignment. Once the fold is set, then they can use the homology modeling and the energy minimization to refine the structure. How to do this? For example, it is a target sequence, right, and these are the different templates. First they take the energy function, right, to align the sequences with the structures, and then they look for the goodness of it, which one will fit with the different templates and align based on the ranks, and finally they get the best one based on the rank. How to use this scoring function? Okay, this is a sequence, they use 3 different aspects. One is what is the probability that 2 residues are in contact. For example, if you know the sequence, what are 2 residues which have high possibility that they are in contact with each other. For example, this lysine and another aspartic acid, they can be close to each other. For example, this and this can be close to each other, or this leucine and this tyrosine can be close to each other. Then see the structures and they have the scoring function, and the second one they see how well they fit with the structural environment. They see the physical chemical properties of amino acids, right, in the sequence, and different secondary structures, right, and the solvent accessibility, whether they are buried or exposed, and look into the structures whether it is buried or exposed or same secondary structures, and based on that they can define the function. Then see the gaps, giving the gap penalty. So, considering these three aspects, they get this total energy. This is the one is the probability of the potential, and this is the structural environment and the gap, and they combine everything and map. If you have this sequence, this would have the energy function of these much values. So, then take all the templates, then which one has the best one, then check all these aspects and rank the models and check, okay, for this sequence this could be the probable model. This is a kind of only 3D model, right, get the sequence and then find the structure. This is called the protein threading based on this fold recognition. So, we discussed about 3 different types of modeling techniques. What are 3 different types of modeling techniques? Homology modeling, ab initio and fold recognition, right. So, now there is a competition called the CASP, right, the critical assessment of structural prediction. So, they conduct once in 2 years, right. So, what they do? So, they get the experimental structures from the crystallographers or from NMR spectroscopy. Before publishing this in the PDB, they keep the structures and give the sequences open to competition. So, you can register and you can try to develop models, or you can use different meta servers. So, there are meta servers available, there you can get the models. With the different models you can choose which among the 50 models which are the best 10, right, you can use that. So, then you submit your model and they evaluate, and once the structure is known they can compare. Because yesterday we discussed about the alignment of structures. So, they use the alignment and see the structurally conserved regions, right, and then see how far your model can fit with the known structures, and accordingly they will tell you, okay, your under this category this method works fine with less RMSD, right. They give different types of proteins with simple proteins or complex structures and so on, right. Then you can also evaluate, okay, which method fits very well with which type of these sequences. For example, if sequence identity is high, we expect the homology modeling work fine, and if other cases ab initio can work, some can do well with the fold recognition and so on, right. So, this is the blind prediction method. Anybody can register and work on that, right, and maybe you can also try, maybe next year we have the competition. So, see whether your method can predict, right, the structures, right, fine with these other available existing methods, right. Summarizing what we discussed today.
Started with structure prediction.
Yeah, mainly with the predicting 3D structures from sequence. What is the name of the problem? Protein folding problem, because comparing the sequences and structures, right, known sequences are around 700 to 800 fold compared to the structures. So, it is very important to build a model, right. Also, structures are important to understand the function. So, then we discussed about the different types of modeling, right. If you the homology modeling, we need to think about see the coverage plus the sequence identity, and then you can choose the template is very important, and then do the alignment corrections, and then do the backbone generation, loop modeling and side chain modeling, and finally optimize your model and validate, right. And finally, if you are happy, you should obey all the rules, mainly stereochemical properties and the energies. And then we discussed about the fold recognition, that is 1D 3D alignment. From the energy functions you can check, identify what are the structures which can fit with your sequence. Then we discussed about the ab initio, so based on the physical energy functions, right. If the homology modeling or the fold recognition fails, then it is very important to do this ab initio. So, you can do with this ab initio structure prediction. In the next classes, I will discuss about some of the applications, mainly how the parameters we derive from the sequence and structures can be used for understanding the protein folding rates or protein stability or the protein interactions, right, and the structure based drug design and so on. Thank you for your kind attention.